Okay. Well, yeah, I'm on. Okay. I have a really bad cold. Okay, so don't get anywhere near me. If I start coughing all over you, turn away. Okay. Just a few updates. Guess we have lab this week. Everybody know where it is? It's in Biology 110 on the first floor, kind of near right next door. So those start today. And so hopefully you'll be doing today interpreting data and graphs. So you'll be looking at kind of similar things to what we've been trying to get you to look at data and graphs, accept or reject hypotheses. You start doing some things in terms of statistical analyses and those sorts of things. The other thing is on Moodle, I've added what's called a course pack, and you'll find the different chapters. Apparently all the quizzes are up there and all the questions. I'm not requiring you to take those, but they might be helpful. They also have some, what do they call, flashcards, or I don't know what do they call them? Flashcards or something? That's not what they call them, but anyway, they're nice in terms of definitions and things like that, where it'll pop up a word and then you turn it over to give you the definition. So that might be helpful to kind of understand certain definitions in each lecture, and maybe I don't define terribly well. So maybe those will be helpful. So what we're going to do today is we're going to start. I didn't email you anything, did I? So I decided to mix things up a little bit. So what we're going to do today, I'm going to start with this example, and I'm going to present an experiment that they did, and then I'm going to ask you to make a prediction about what you think should happen. And what I'd like you to do is to work together in groups of 3 to 4. You can turn the desks around and you can talk to one another. And what I'd like you to do, I'm going to time you. I'm going to give you 3 minutes to answer the question and write it up. Hopefully that'll be enough time, okay? Maybe 3 to 5, okay? And how about you want some bonus points when you do that on your first exam? Okay, so this is kind of what I do to mix things up a little bit. I might do it at the beginning of class, I might do it in the middle of class, I might do it at the end of class, but we'll do this one at the beginning of the class. What we're going to do at the end of this class, I'm going to not do the last 2 slides in phylogenetics. I'm going to do a really short, like 5-minute recording. At the beginning of class next time, we're going to do a little iClicker question based on that little video that I did. How's that sound? Mix it up a little bit. Okay. So here is this. This is what's called, where we're going to be spending a whole chapter on life history evolution. And what life history evolution looks at, how do organisms develop patterns of reproduction in terms of the size and number of offspring that they reproduce, when they start reproducing, like at what age, earlier versus later. And then another key thing we can look at is how much reproductive effort does, for example, a female put into reproduction, right? Is it a lot or is it less? And it's always relative. It's always compared to another population. So we're going to do a little exercise here. And remember we looked at these Trinidad guppies. And some of them live in populations that have what's called a high predation site with these pretty voracious cichlids. And these are things that they're pretty voracious, but they primarily feed on adult guppies. In the upstream sites, higher up in the mountains, in the upper, like the headwaters of the streams and rivers, there's another predator, Rivulus, a little smaller. It's not as voracious. It's a pretty good predator, but it specializes on juvenile or smaller guppies. And so these even in the same stream, they have very different kinds of patterns of predation on what kind of guppies they feed on. And so one of the first patterns that we saw is that in the high predation sites and the low stream sites, they produce a large number of offspring that are relatively small. So this is always a larger number and smaller. Okay? In the low predation height upstream, they produce smaller but larger offspring. Okay? So when you think about that, organisms have a certain amount of energy that might be available to reproduce, how many eggs they produce and how many offspring they produce. So given like a fixed amount, you can, it's like a pie, right? You can make a lot of small individual pieces, right? Or you can make larger, fewer ones, okay? And so we're going to spend a lot more time on this sort of concept and idea, but that's the basic idea why you see that trade-off between size and number. That if you produce a lot of offspring, right, you in general can produce smaller offspring. If you produce fewer, they're in general going to be larger. Okay? So when you think about that, let's just look at the particular example on the upstream site. So when you look at this upstream site, what they're producing are relatively fewer offspring, but much larger. why do you think, why, and what we're going to say is natural selection is favored producing fewer but larger offspring. Can you come up with a hypothesis? This is not the one we're going to develop or ask you about. Why do you think that kind of pattern is favored in that kind of environment? Any ideas? Why, huh? Yeah, so the basic idea here is that maybe it's better for a female to produce larger offspring. So they're viviparous, so they produce offspring inside them and then they give birth to them live. And so they're producing larger offspring. The idea here is natural selection probably favors that because that kind of gives them they can grow faster and more quickly and escape predation that way is one of the hypotheses why that might be favored. So maybe it's better to produce larger offspring and they might be less susceptible to predation by that thing, or they might escape that period where they're prone to predation much faster. Does that make sense? Okay. So that's what happens in terms of size and number of offspring. So let's look at another life history trait. So what I'm going to ask you, and I'd like you to work in groups, write a piece of paper, write the answer, and tell me in one sentence why. Don't go on for, half a page, if you can. Okay? So what I'm asking here, this was a great an experiment that was done. They took guppies from the lower reaches, and there was a pond or a stream bed up here that was isolated, basically had no guppies in it, but had some rivulets in it. So they took females from here, from the high predation site where adult predation was common on the guppies, and they relocated them up here where they were experiencing the rivulous predation. Okay? So guppies from the high predation site were transferred to the low predation site. So they went from a cichlid predation to that rivulous predation. So here's the question. Should adult female guppies with a lower risk of predation up in here, right? Remember those rivulets don't eat adults as much? Should they invest more or less in reproduction compared to the downstream females? So it's 50-50 shot, right? So does selection favor greater reproductive effort or less reproductive effort? And how might you explain that in terms of natural selection? Okay? So what I want you to do, I know this is hard, but what I want you to do is talk to your neighbor and discuss however you want to do groups of 3, 4, however you want to do it. And so I'll give you 4 minutes. Okay? Yeah, and you're going to turn it in. Turn in the sheet with everybody's one sheet with everybody's name on it. One bonus point on your first exam. Well, try to get it right. I'll give you a 2-minute warning. I won't answer any questions. No, it's kind of how much energy they put into reproduction relative to the other population. Do they put more or less in general? It's kind of tough. Yeah, join any group. Okay, 2-minute warning. Make sure everybody's name is on the paper. And 35 seconds. Yeah, so just yeah.
Can I do it and then like turn it in? I don't think anyone feel comfortable with me just writing my name on, because they're pretty much done.
Okay. Just send me an email.
Okay.
I'm going to give you the answer, so just tell me who you are, and I'll just kind of, yeah. Okay, time up. Okay, just pass them forward. I'm real generous with extra credit, so. Okay, I'll start talking about the answer, even. So, how many people voted for more? Okay, how many people voted for less? Okay, less is right. And this is a tough, I'm telling you, this is a tough one. And I haven't discussed life history, evolution, and a lot of things, but here's the basic idea, and this is what happened. When they did this transplant study, what they found is they started to produce fewer, larger offspring, and the amount of energy they allocated to reproduction in those upstream sites was a lot less. So does somebody want to tell me, the people who said less, what was the reason? Or are you afraid? Okay, yeah. Would you? No, the amount. Right. And I should have explained that a little better. So it's basically saying out of the amount of energy, basically when you think about life history, an organism it can divert energy into reproduction, growth, and maintenance. And the question is, do you divert more into reproduction and less into growth and maintenance? That's probably, okay, that's fine. So now, if you had to believe that, now do you understand why it's less? The basic idea is pretty simple. They have a higher those females in that upstream site actually survive a much longer period of time once they reach adulthood and reproduce multiple times. And so the idea is they just invest less in each reproductive event because they have a high probability of surviving throughout a much longer period. Whereas if you're in that downstream site, do you think you want to stick around and just invest a little bit in reproduction, right? Probably not a good strategy because what's your likelihood of getting eaten? It's pretty high. So the idea there, where you have a high risk of dying as an adult, right, you should really put all your energy into reproduction, not growth and maintenance. Does that make sense? Okay. I'll give you all a point. Okay. So anyway, I think these are some of the things we'll try. Any questions about that? Does that make, I probably should have phrased it a little differently as I was reading. I said, I should have phrased it differently. But okay, that's how teachers are. Okay. So again, does that make sense? So we'll try to do this on a fair, at least, if I get too far behind in my material, which I always do, I got to pound, I got to pound the material into you, right, and give you as much, more material than you can master for each exam, right? I'm kidding. I don't like that approach. Okay, I'd much rather have you learn. Okay, so let's finish up this lecture, and then we're going to move on to phylogenetic trees. And the last thing we'll talk about are phylogenetic. Did you have a question? Okay. So when we've been talking a lot about adaptations, that natural selection favors certain kinds of traits, or what we call suites of traits, because they confer a higher survivorship or a higher reproductive rate of the individuals or genotypes that possess them. And these are what we call adaptations. And we'll be spending a lot of time on those. So one of the other things we also understand really well is that adaptations always have limits or constraints. So these are pretty dramatically different kinds of birds. And what's different about an owl and an ostrich? Size, okay? What's different about the way they make a living, too? Their eyes, okay? And these eyes probably have something to do with what they eat or how they're eaten, okay? So when you think about this, when you look at owls, and along here is the total field of vision versus the range of binocular vision. When you look at these things, ostriches have a huge total field of vision, right? Almost, not quite 360 degrees, but, and how are their eyes situated in their head? They're kind of on their side, right? Whereas owls, are they situated on the side and oriented more that way? They're much more, or their total field of vision is a lot less, but their range of binocular vision is much higher, right? That is, so why do you think that is? Why is this sort of high range of monocular vision, minimal binocular vision important for ostriches and less so for owls? Yeah. Huh? Because owls fly, and what are they doing when they're flying? What is their ultimate goal? They're a predator, right? And so they're feeding on, in general, mice, on rodents, sorts of things, and so vision, directed binocular vision, being able to easily locate prey. A lot of them use sound. Don't get me wrong, but this really focused binocular vision is really critical. What's the concern of an ostrich? Sort of monocular vision, why is it that way? What's coming after it, potentially, is one of the big issues, right? Is they want to have this wide range of vision to potentially, alert them to predators and things like that. They do have some binocular vision, and some of them, they really don't need that sort of really wide binocular vision because they eat on, I should have looked this up, ostriches, probably lots of grains, insects, and things like that. And they can just look really quickly and make a jab at it, and they don't really need this wide binocular vision, okay? And the idea here is that, if you have selection favoring binocular vision, it comes at some cost or trade-off in terms of monocular vision, right? So this is one of the ideas that adaptations, you can't really produce an organism that has huge binocular vision and huge monocular vision. There's a real constraint here. And adaptations really always have limits or constraints, and something we always want to understand. Okay? I'm not going to spend a whole lot of time on this because we're going to move right into phylogenetic trees, but one of the key things that phylogenetic trees allow us to do is to understand how often fairly complex traits arise, and potentially we can argue what are the selective mechanisms that led to the origin of traits. So I'm not going to spend a whole lot of time, and your book does a really nice job on this, but this is a phylogenetic tree, primarily birds, its related ancestors, and a whole series of other extinct relatives. what we do here is this is a phylogenetic tree. On the tips these are genera, a whole range of modern birds. Modern birds and Archaeopteryx are sister taxa in the phylogenetic tree. And what we mapped on here is what we said at this common ancestor, at this blue thing right here, right preceding to it, is when we envisioned feathers originated. Okay? So what do you think is true about all of these things that are branching upward from this ancestral node? Well, they all have feathers. So we envision and we're saying that that's probably in that putative common ancestor, all those different kinds of organisms, all in here, they all had feathers. Okay? Now, why do you think feathers arose? Can you think of some explanations for why natural selection might have favored the evolution of feathers? Flight, that's an obvious one. Can you think of anything else? Yep. One of the options probably would be protect the skin from drying out or desiccation. That's a good one. Can you think of anything else that feathers might do? Yeah. Huh? Yeah, and what do you think, what kind of properties might feathers have in what kind of environment that might be an advantage? Cold, okay? So one of the things is they probably have great insulative properties too, so that they might prevent body heat from dissipating, in cold weather and things like that. So in your book, they outlined about 8 possible reasons why feathers might have evolved, and they may not all be mutually exclusive, okay? So, and then obviously, feathers, do you think, didn't originate for flight, did they? No, because, and this is all we're going to talk about here, this is when modern birds, Archaeopteryx, and this Microraptor had the evolution. Obviously, there were a lot of things beyond feathers, but the forelimbs and things like that were modified, and a lot of these the origin of gliding flight originated here, and there were huge modifications in terms of probably what those feathers look like, how they're really critical for flight, and all those sorts of things that occurred in that, and especially in modern birds, the evolution of incredible feather structures, unique feather structures that really enhance flight and all those sorts of things. Okay, so what we would say is that it's obviously the common ancestor to modern birds, Archaeopteryx and Microraptors, where the gliding flight originated, and probably a lot of modifications of those feathers and things occurred there. But feathers did not originate probably originally for flight. And your book goes into all these things about exaptations, adaptations. I'm not too worried about you understanding that sort of language. Okay? So the key thing here is phylogenetic trees tell us these are fairly complex structures, right? Feathers. And how they've evolved and how they've changed, but they don't arise out of the blue or de novo, right? The feathers originated probably for other reasons, then they were modified to enhance flight and gliding flight and all those sorts of things. So often what we call complex traits don't arise out of the blue, but are traits that are modified to new functions, okay? And there's all stuff about eyes and vision and things like that, but okay. Okay, there are some things, you can all, when you leave, you guys can go all have coffee, right? Or sit out in the quad and discuss these things. Right? Okay. So anyway, those are some of the things as you reflect back, these are the key points that I really want you guys to kind of take home from this lecture. Okay. Okay, where am I? Okay, lecture 3. Lecture 3. So what we're going to do, any questions about that lecture or the wrap up on that previous? Okay. Now we're moving on to chapter 4, phylogeny and evolutionary history. I don't want you to do the inquisitive module for phylogenetic trees quite yet, right? I want you to do that in the second part of the course, because I'm going to come back to chapter 5, which is how we actually construct phylogenetic trees. And that'll be later, and you have a lab in the second part of the semester on basically creating phylogenetic trees from some data you have in the lab. So I'm going to hold off on doing that inquisitive module, do the other 2 first, and those will be extra bonus points for the first exam. Okay. I have to make the exam really hard if I give out a lot of bonus points, right? Okay. Just kidding. Okay. So we're going to talk about phylogeny and evolutionary history. Here are, the sort of questions and goals of this particular lecture. How do we read and interpret a phylogenetic tree? How do phylogenetic trees help us classify the diversity of life on Earth? How do phylogenetic trees help us understand the evolutionary origin of similarities? And we're going to distinguish what we call homologous similarities and analogous similarities and the difference between species. And then how do we map characters and traits onto phylogenetic trees and test hypotheses about evolutionary events? And the last thing I'll do, that's going to be the short video. It'll be a 3 to 5 minute video I'll post on Moodle right after class, and then there'll be an iClicker question at the beginning of the next class. Are the last 2 slides, where I talk about these cool snake venom delivery systems and how they, kind of an interesting story. So phylogeny, when we think about it, can be represented at a lot of different scales. So there's a huge interest right now in actually reconstructing entirely the tree of life. And that is, it's been a long time since I took the introductory biology, okay? But these high-level classification schemes from bacteria, archaea, and eukaryotes, the 3 major groups of organisms, right? That's a very high level, right? When you think about that, what do we call those now? Kingdoms? Okay, so those are the 3 major kingdoms. Some people think there are more, okay? There's some argument about that. But when you think about that, this is a very high-level phylogeny. And you can see that there are eukaryotes in here, and you can see that out of this, you have all sorts of plants, all sorts of other things. And here, what's this little red branch right here? It's hard to see, but it's animals, okay? And you can obviously say, well, there's a lot of other things in, so this is a very high-level phylogeny, right? There's obviously a lot of diversification from the common ancestor to animals, and I can't even read what it is, right? But there's been a huge diversification of animals and those sorts of things that aren't even depicted on this tree. It's just a small single branch. And so this would be a high-level phylogeny. Here's another one, including a fair number of vertebrates, from crocodiles and birds to turtles to snakes to mammals, and these are kind of the phylogenetic relationships among what we call tetrapods. Not all of these are tetrapods. And so this is at a finer scale than this one. And then you can even go down to population level phylogenies, and that's a lot of what I do. I'm sort of interested in the sort of phylogenetic relationships among populations and how they're related, how populations become differentiated and isolated from one another. So there's a lot of interest in this. This is an example of Mycobacterium. This is an important pathogen in a lot of human populations. And so here are a group, it's hard to see, but each one of these clades are different colors. Here's kind of a bluish clade, a slightly different clade. Here's another reddish clade. But each one of these represents kind of geographic area throughout the world where you find these populations that group together. So you find East African Mycobacterium sharing a common ancestor, right? You find India and East Africa sort of sharing a common ancestor, and they all come back to that common ancestor. And then you have, what do you have? Europe, America, and Africa. So you have these sort of different populations that form these groups that share a common ancestor. So remember again, what we have out here, what do we call these of the phylogenetic tree? These are the tips, right? And so these are the things, and then down here are the branches leading to common ancestors at each one of these nodes. Okay, so phylogenies we can look at this at a lot of different levels and at a lot of different scales. So how do we do this? And again, we're not going to spend any time on how we got, at least right now, from here to here. That's what we'll be doing later in the course and in lab. But what I can tell you is what we can do is we can construct trees with different kinds of data. It can be DNA sequence data. It can be morphological data. It could be behavioral data, a lot of the things we talked about, that you can reconstruct sort of the phylogenetic relationships. So here's just an example. What are these things right here? Yeah, that's a DNA sequence, maybe of a particular gene or a group of genes. For, this could be individuals from population one, population 1, population 2. These could be species. These could be genera. They could be whatever. They could be any taxonomic level you wanted them to be. So from these data, and this is what a lot of people are doing right now, is primarily given that we can basically sequence genomes fairly rapidly, fairly cheaply. There's a lot of interest in using a lot of DNA sequence data to do phylogenetic trees right now. So what we can do is from this, and I'm not showing how, you can infer the phylogenetic relationships from DNA sequences. So what this basically, fundamentally, you can say is probably you're getting groupings of these taxa because they probably are, in some sense, more similar to one another, and we'll talk about how that similarity arises. But what you can see from here is what are the, so 2 and 3, right? What you can see is they pretty much have the same DNA sequence except for one base pair, and what you're missing, there are a lot more than just 7 base pairs that were used to reconstruct this tree, so we're not looking at all of them. But what it's saying is 2 and 3, what are we saying about those given this phylogenetic tree? They're more closely related to one another, right? They share a more recent common ancestor with one another than they do with any other species or population in the phylogenetic tree. What's another example of a group of what we call sister taxa, especially at the tips of the tree? 5 and 6, right? So those two tend to, and again, you'll see, and this isn't all the data, only one base pair difference, but don't go in and look at that and try to reconstruct it because it won't work, okay? And then obviously we have all these sorts of things. And then so what are we saying about that node right there? That is the common ancestor, or the branch to the common ancestor of what species? 2, 3, and 4. So 2, 3, and 4 share a more recent common ancestor with one another, and those sorts of things. And here's the root of the entire tree. This is the ancestral node to all the taxa, and those sorts of things. Okay. The things I'm kind of more interested in right now is how do you map traits onto these trees and then try to figure out when do these traits originate, and then we might be able to understand what were the selective mechanisms that led to this. Okay. So take this tree, and now what we put up here, instead of the numbers, what have we put up here? Yeah, those are flowers that are, we assume, representative of each species. What's different about all these flowers up here? What's kind of, yeah. So one has, there are 2 traits that we can map onto this tree. One is shape of the flower, and what's the other one? Color. Okay, so let's look at this. So when you look at this, the origin of a new shape, these 3 species obviously have the same shape. Those 3 have a different shape. So where would you branch, where would you say that new trait originated in terms of the new shape? Right at the common ancestor to those 3 species that have that trait, okay? Do you need to, so basically what you're saying is, what's the ancestral state of flower shape in this group of organisms? Whatever that one, a kind of a rosette sort of flower, or that sort of flower, okay? So what you would say is this shape right here is the ancestral character state, and it evolved to a new state in the common ancestor of those 3, let's just say 3 species. Where's the origin of new coloration in this tree? All of these are white, right? And that has an orange coloration. So where would you, what's the ancestral condition flower color in this phylogenetic tree? White, okay? At the base here, and where did it evolve a new coloration? In the branch leading to that other species, okay? So that's how we're going to be, you'll be saying to yourself by the end of this, too much time on this topic, okay? So we'll come back to that, and we're going to be looking at examples of how we map traits onto trees. And when you think about it, that orange coloration, what do you think happened there? If you were interested in, why did that evolve a new coloration? What might be the first thing you'd look for? Huh? Yeah, a mutation. Okay, so let's hope that there's a genetic mutation that led to a new coloration. And then how might it be favored by it may have been favored by natural selection, and what do you think it might have something to do with? Probably pollination. And perhaps it's a new pollinator, right? Where certain kinds of colors are more important in terms of recognition or effective pollen delivery. That those could be some. So then you can go in and look at all the pollinators of all those things and say, here's, and everybody knows what kind of pollinators like orange plants, right? Okay. So let's go back. There are lots of ways we can depict phylogenetic trees. I, in general, like this one, but you can present it any way. One is what's called a tree, or you can do a ladder. I can't remember how they do it in the lab. It doesn't really matter. These two things depict the exact same thing, and the important thing to recognize is at the base of all trees or ladders is what we call the root. Okay? It's like a tree, which is the common ancestor to all the taxa in the phylogenetic tree. And obviously time, this is ancestral, and this is more recent. So time is moving. As you go through this tree or ladder, time is in this direction. Okay? I've already talked about this. Here we have the interior nodes. All of these are interior nodes. These are exactly in the same place. It's just depicted differently. And so you have interior nodes, multiple interior nodes that are a common ancestor to all those groups of taxa. And we just refer to these as interior nodes. And here are the roots, and these are the tips. And yes, crocodiles and birds share a more recent common ancestor with one another. Okay. So again, interior nodes of a tree represent what we call common ancestors. And given this has a time dimension, right, from current to deeper or more ancestral, so time this going up this way, time going down that way, deeper nodes represent older common ancestors. So here, for example, here's a hypothetical common ancestor to species 8 and 9. There's a hypothetical common ancestor to 6, 7, 8, and 9. Why does this thing keep turning off? Okay. Which is the deeper node and represents a more older common ancestor? The blue one, right? These are kind of analogous in some sense. And this is, of course, the deepest, most ancestral node on the tree. This is nothing new. You had this all in, did you have this in biodiversity? Yes? Okay. I'm not too worried. You can rotate any node on a tree without changing the structure of the tree. That is, by rotating a node, nothing changes in terms of the sort of common ancestors and descendants, the patterns within a particular node. So let's look at just rotating on this node. Here in this tree, you have 3 and 4, just as a sister taxa. If you rotate that, you put 4 over here or 3 over here, does that really change anything? It doesn't change anything. 3 and 4 share a more recent common ancestor. You just put one on the left and one on the right. It's meaningless. It has no effect on the structure of the tree. If you go down here, you can put 4, 3, and 2, and again, nothing really changes, right? The original way to do it was 2, 3, and 4, but if you rotate on that node, you have 2 over here, but 4 and 3 still share a more recent common ancestor with one, much like here. And do they all share a common ancestor at that node still? So I'm not going to belabor this point. It's really not interesting. So you can rotate any node on a tree without changing the structure on a tree. Here's some. So one of the big controversies that occurs when people start reconstructing phylogenetic relationships among organisms, depending on what kind of data they use, you can often get sort of different trees depending on what kind of data you use. And this is considered kind of one of the more classic ones where you're looking at, this is an example in this phylogeny, placental mammals and marsupial mammals in this particular tree are considered sister taxa, okay? And the monotremes, and where are the monotremes and marsupials found? Australia, right? And placentals are primarily widespread primarily outside of Australia, include, just as an example, elephants, but obviously we're placentals too, okay? So this particular tree found that placentals and marsupials share a more common ancestor with one another. Another piece of data, in this phylogeny, marsupials and monotremes share a more recent common ancestor with one another, and then placentals are outside of it. All of them are basically still, come down to a common ancestor. So a lot of times you can get sort of uncertainty about evolutionary relationships depending on the kind of data you have. So here you can represent this. These two data sets present different scenarios or different relationships, and uncertainty about relationships can be represented by what's called a polytomy. Maybe that's in your flashcards. But what do you think what's a polytomy? So basically what you have is you don't have any clear, you basically have all three taxa kind of coming back or coalescing to a single ancestral node, and you can't determine what you're saying here is the two data sets conflict, and we can't make any judgment about who is more closely related to whom. Should we give up? No. What we should do is collect more data to try to resolve that problem, and the problem has been resolved. Guess which one of these is, well, some people would argue with me, or with other people. Which of these data do you think is from DNA sequence data? It's hard to say, but the first one. Turns out the first one, large-scale DNA sequence data, even more beyond this, suggests that placental mammals and marsupials share a more recent common ancestor with one another. And it's the morphology data that led us to group marsupials and monotremes together, and in fact, that's probably wrong. Okay? And the DNA sequence data strongly support that placentals and marsupials are more closely related to one another. So again, we can have controversy, but data can always sort of help resolve these sorts of things. So here's flashcards, okay? And again, I don't give multiple choice questions on exams that usually say a monophyletic clade is. I don't usually do that kind of sort of thing, but you kind of have to understand the language and the definition at some level to understand it, but I'm not going to test you on like definition sort of things. And I'll give you examples of my exams so you kind of, and they're probably already out in the cloud somewhere. Are they? No? Okay. They should be. Okay. So let me give you an example. It turns out that some taxonomic, the nomenclature of various kind of groups of organisms suggests that elephants, rhinoceroses, and hippopotamuses are often classified together with one another. And it turns out that's entirely wrong. So what I'm showing you here is this is a group of taxa, pretty high level, ranging from pigs to ruminants to hippopotamus to cetaceans, whales, horses, rhinoceros, tapirs, hyraxes, manatees, and elephants. So what we have represented here, each one of these colors kind of represents what we call clades that include all the taxa that share a more recent common ancestor are called monophyletic clades. So how many monophyletic clades in general, how many monophyletic clades do you see on this tree? Yeah, it depends how you do it. 3 or 4 is how I'd present it. Here's one, right? Here's kind of the bigger one in the lighter blue, but then there's a little monophyletic clade in here, but I don't know why they did this. Anyway, let's just say 3. So these are the 3 major monophyletic clades right here. And that is, they all share a more recent common ancestor with one another, and they all go back to that sort of single ancestral node, and they're the common descendants from that ancestor. So classifying elephants, rhinoceroses, and hippos together, is that right or wrong based on if this is the real tree? Should we change our classification, how we group them into particular kinds of taxonomic units? Yeah, we should. Because it turns out elephants are up here, rhinoceroses are up here, and hippopotamuses are down there. They're in different monophyletic groups. So we probably should say, oh, this is a class of organisms, right? taxonomic level. Oh, this is a class of, or however it is. And then this might be a class or whatever, order. I forgot. Okay, it's been a long time since I took introductory biology. Okay, so these things do not share a common ancestor. And what's called that classification scheme, and again, I'm not going to hold you to a lot of this. That taxonomic classification scheme is what we call a polyphyletic classification scheme. It includes, they do not share a common ancestor, and that's bad. We want to classify things because they share common ancestors and they're monophyletic. That is, they group back to a single common ancestor, and that's how we would classify these things taxonomically. And there's been a lot of revision of our old classification schemes based on DNA sequence data and a lot of changes. Let me just, I'm not going into paraphyletics, so we're just going to ignore that. I don't want to burden you with too many of these buzzwords. But again, here's an example, tetrapod vertebrates, right? From amphibians, mammals, snakes, turtles, crocodilians, and birds. Tetrapod vertebrates we consider a monophyletic group. And we might consider tetrapods to be a good way to classify them at a certain taxonomic level because they share a common ancestor right here, and this is the common ancestor to all tetrapod vertebrates, okay? Fish are a problem. We're not going to worry. It turns out we shouldn't classify fish the way we've classified them, but we're not going to worry about that. There also are other ways we can kind of look at trees. I prefer this, but we have some things that are called cladograms and some are called phylograms. And what you'll notice here, what's different kind of about these two trees, especially over in here at the tips of the trees. What do you notice that's different? Yeah, it's a line, and they're all at the same point. They're all basically come to the exact same point. They're right justified, okay? So humans, chimpanzees, bonobo, gorilla, orangutans, basically these branches all go to the exact same point. So this suggests what this kind of cladogram doesn't tell you anything about the evolutionary changes that have occurred along branches or anything like that. These are what we call cladograms, and they're perfectly fine. It suggests that humans, chimpanzees, and bonobos share a more recent common ancestor with one another, gorillas, and that sort of thing. And actually, from DNA sequence data, you can actually create a phylogram. It's easy to do. And here's an example. This is for a phylogram. What's different? You'll see each one of these DNA sequences of a variant of SIV or HIV. It shows up at a different part of the right hand of the branch. But what you can see is these branches all have different lengths, and they vary. And so what this is saying is, basically what this is saying, these nucleotide sequences, it's a measure of the amount of sequence divergence along these branches. And let me just give you kind of one cool example. Just going down and looking at these two isolates that they sequenced of HIV, really easy to do. Here's HIV-2b, HIV-2a. They share a common ancestor. This one, you'll see, has a longer branch along there compared to that one. And so what that shows is that more changes have occurred along that branch leading to that isolate than that one, okay? And we can, what might that mean, do you think? Well, it means that this isolate has accumulated a lot more changes or more mutations from the common ancestor compared to that one. It's all it's basically saying. Yep. So you can see, yep. So what this means is, think about it, that's the common ancestor. You go up this branch, those are both at the same point, right? What that says is there have been more changes along that branch leading to that B isolate of HIV, and this is much shorter. And you can see a few other examples of this, but it turns out that particular isolate turns out to be in the original explosion of the HIV and AIDS epidemic. That's the one that was actually the most virulent and the most nasty one. So there were some changes associated with it along that branch that led to a really high, a strong effect on humans who had HIV. So here's another great thing you can do with phylogenetic trees, especially from sequence data. And again, I'm not going to ask you to understand all of this, but this is an example of orchids. Does anybody like flowers? Orchids are nice, aren't they? Right? They're beautiful. So, and there's an incredible amount of diversity in tropical orchid. They're primarily tropical. Incredible diversity throughout the tropics. So we know from DNA sequence data, from dating, and again, we're not getting into how we do that. We'll eventually get into how we do that, but we can sequence trees. We can sequence organisms, lots of species, lots of individuals within species, and we can reconstruct the phylogenetic relations. We can actually date the time at which orchids originated. And here's the common ancestor right here in orange. And along here is sort of the timing. And so this is 75 million years ago, 50 million years ago, 25 million years ago to 0. Is this a cladogram or a phylogram? Yes, it's a cladogram. Okay. I was about ready to say a phylogram, and that tells you how I'm thinking today. Okay. Yep. This is a cladogram. They're all in the same point. There are no differences in branch lengths. So here, the DNA sequence data suggested that orchids originated probably 80 to 85 million years ago. Something happened about 65 million years ago. Anybody know? Yeah, dinosaurs. And then what happened to them? Right. So at the KT boundary, there was a huge asteroid event, right, that caused huge widespread extinction, right, in lots of different taxa and organisms and those sorts of things. So again, we're not showing that. But this is kind of the KT boundary right here, where there was that mass extinction event due to the asteroid, and all the plumes and incredible climate change associated with a lot of death and extinction. Okay. And then these are kind of different kinds of orchids. And these are probably families. I believe these are families of orchids. Here's the KP. Oh, I thought it was KT. I'm confused. Okay, anyway, so when the extinction event occurred, what does each one of these things represent? What does this show you? What this suggests here is there are certain kinds of things that have kind of the width of that thing at the end. What do you think that's telling you? Yeah, or diversity, right? Abundance or diversity. And it's mostly at the genus level. So it's how many genera there are within each one of these families. So what does it suggest? Do these families of orchids differ in terms of their diversification rates after that boundary? What you can see is, did that diversify much? This is basically an old ancestral clade of orchids, right, that has persisted through that event and probably hasn't diversified at all. These a little more, see how it flares out a little more, and basically the width of this thing right here as you go along from 65 million years to present is the kind of the diversity you see in the number of genera. And these two, and especially this one, shows an incredible explosion of diversity, especially it's ongoing, but from this period. And this is, these two clades showed incredible diversification, high rates of speciation, and those sorts of things. So one of the things we might ask ourselves is, why do those have low rates of diversification or no diversification, whereas why was there an explosion here? Any ideas? Oh, good. Okay. That's just, you can do a lot with phylogenetic trees. So the other thing we're going to look at are opsins. Anybody know what opsins are? Things in your eyes. They're visual pigments that basically facilitate color vision. They're different kinds that we call, primarily I'm looking for the right word. Results in sort of absorption at short wavelengths, medium lengths, and long wavelengths. So these are the different kinds of opsins that organisms with color vision have. You can have a whole set of different kinds of opsins. They kind of fall into these sort of short, medium, and long. And so then they kind of allow you to have sort of assess blue, green, and sort of orange pigments that allow you to see those sorts of things. So again, we know a lot about the different kinds of opsins at pretty high levels in terms of what organisms have. And so what we find is, we'll come back to this, is that here's a phylogenetic tree. This is a pretty broad one. Along here represents the kinds of opsins that they have. So squamate reptiles and birds have 4 opsins, ranging from the kind of purplish to the more yellowish orange at this different wavelengths, okay? So what we can do is we can kind of, given this phylogenetic tree, map the changes in the opsin loss and gains of these various kinds of visual pigments. And let's just kind of go through this. So this is what we have at present, right? In rodents, New World primates, baboons, and humans. We know this for a lot of different organisms. We're just showing baboons and humans together. So what we're saying here is, we'll come back to this. This is often what we call the group that we might use, what we call an outgroup, and allows us to determine what the ancestor was to all these things. But what we're saying here in this particular phylogenetic tree is the ancestor, tetrapod ancestor, probably had all of these 4 opsin pigments, all of them along here. So you go up here. Did anything change along that branch? Nope, they all have 4. So nothing occurred here. We then go to this branch that bifurcates or splits here, and what happened? You lose 2. And the way that works is fairly simple. You'll be doing this a lot in lab, is when you look at rodents, what do they have? They have 2. And what do the New World primates have? They have 2. So basically what you're saying is that what happened along that branch that led to rodents and then the common ancestor to all of these, to especially the New World primates, what were lost? Well, I can't even see. That one, and which one? That, yeah. Yeah. So they lost that one and that one, okay? And then what happened in the, they lost it. Then what happened in the common ancestor to baboons and humans? They gained the green visual opsin, okay? So that's how we can kind of use these trees, and then we can obviously say, we can map these changes, and we might ask ourselves, why were the opsin genes lost in the lineage leading to early mammals? And let's just, what we're talking about is, why do you think those kinds of visual pigments were lost? Do you think it was just by chance? Yeah, because they didn't need it, probably. And what do you think was unusual about those sort of early mammals, especially compared to baboons and humans? And the New World primates is another, I'm not going to get into that. What do you think? Why didn't they need those pigments? They're nocturnal, great, okay? So one of the arguments is that these particular kinds of opsins are really not needed, especially for nocturnal vision, so there's no selection to maintain them, and they were lost, okay? Obviously, is that true in us, in baboons, and all sorts of other organisms? And this is more than just baboons and humans, but certain opsins are very important for diurnal and color vision. Okay. Let's talk about homology and analogy. Remember what that is? What's a homologous trait? Same traits, but what causes those 2 species to have the same trait? They share a common ancestor, right? So that's what we call homology. That is similarity among organisms because of shared common ancestry. What's another way that organisms can share traits in common? Yeah, through adaptations or, what we call convergent evolution, right? Unrelated organisms can often evolve similarity, right, due to natural selection. It has nothing to do with common ancestry. And so that's what we call analogy, or convergent evolution. And we'll look at a couple examples of those. So here's a kind of a classic example of this. So wings are homologous. We could consider wings as homologous traits in butterflies and true flies because they arose in the common ancestor to all Lepidoptera and flies, and these all don't have it. So we say that wings originated, and they're homologous and they occur. Do these wings look exactly alike? No. Obviously, the wings originated here, but the diversification, shapes, colors, all those things undergo a lot of diversification very differently in Lepidoptera than they do in flies, okay? But we're just talking about how they originated and do they share a common ancestor. Here's another example. Adaptations for hovering to feed from flowers are what we might often call analogous traits. Here's a hummingbird. Do you know what this is? That's a hawk moth that is very similar, feeds on flowers with really long corollas. They're nectar feeders. They're primarily nocturnal, and they obviously have a mouth part that goes deep into these things, and that's what they feed upon. But obviously, would you say these sort of wing shapes and the hovering, are they similar between those two because they share a common ancestor? No. The adaptations for hovering to feed from flowers, whatever these adaptations are, right, how they flutter their wings, the rate at which they flutter their wings, shape of wings, all those sorts of things, they're similar, not due to common ancestry, but because of a similar selective environment. That is, they have to hover to feed on flowers. And so it led to similarity, but not due to common ancestor. So where would you put the adaptations to hovering? Where would you put the branch on these trees? Where would you put that branch on the trees? Would you put it down here? Yeah, put it on each one. I need to actually put it on probably the hummingbird lineage, right? So you put it there, and then you put it on here. It's kind of two independent origins due to selection. Okay, iClicker question. You ready? Maybe. So let me just read it first. Homologous traits are found in two or more species because some evolutionary process, usually natural selection, has independently fashioned similar traits in each species, A. B, those traits have been inherited from a common ancestor. C, hybridization between the species passes the trait on to both, and evolution by natural selection requires, what did I say? Requires heritable variation. I haven't turned it on. Hold on a second. So A, B, C, or D. What am I doing? Okay. I'll post these on Moodle too at some point. Okay, everybody in? Remember, this counts for attendance. Yeah, this just kind of counts for attendance and help reinforce. Okay, everybody in? What's the answer? B. That was really easy, right? Was that easy? Let's see. Yep, Okay, good. That was easy. So remember some evolution has independently that would be analogy. Okay, so kind of keep the homology and analogy separate. So that was pretty straightforward. Okay. So I'm really an effective lecturer, in other words. Or I'm just kidding. Okay. So let's stop. And I am, let me see where we are. I'm going to stop there. Well, let me go through this one. Let me just finish this one. Here's an example of convergent evolution. This is pretty similar to what we said in the New Mexico desert. This is a woman at Berkeley that's been studying these where shared coloration is not due to common ancestors but natural selection. So here you have sand dunes, grasslands, and lava fields through all kinds of organisms. Here's the habitat. Here are fence lizards and mice. You can see, are fence lizards and pocket mice very closely related to one another? Not at all. They've converged on very similar, not exactly the same, but very light coloration in sand dunes. In grasslands, a little darker, a little better matching. And then you go to the lava fields, you find much more melanic, darker in these two very unrelated organisms. And so we would argue, again, probably what's leading to that? Camouflage favoring those kinds of coloration patterns in those very different kinds of environments, and that those evolved through natural selection and they're analogous, not homologous. Okay, let me just tell you what I'm going to do. I'm going to video record this lecture really quickly, okay? And so if you can review this, I'm not going to ask you any questions about this part, but take a look at those things, and I'll go through that. I'm going to talk about this. So what I want you to do is look at this and read in the book if you've got it, and this. And we're going to discuss those two things. I'll have an iClicker question at the beginning of class that focuses on that incredible venom delivery system and how it evolved in these kinds of snakes. So that'll be the first iClicker question at the beginning. So try to get to class. I'll probably do another one in the middle. So, okay.